That's why you left it off.
So, today we're going to talk about the respiratory system. The CLOs. The objective: explain the process of inspiration, explain the process of expiration, describe all the lung volume and lung capacities. Make sure this is very important, okay? Not saying that other stuff is not important, but this is important. Explain the following terms and disturbance: metabolic acidosis, metabolic alkalosis, respiratory acidosis, respiratory alkalosis. Okay, so these are the main themes that we're talking about today. Okay. Now, there are two types of pneumocytes, type 1 and type 2, and they both have different functions. Type 1 pneumocyte is your squamous epithelium. This is the cell that is actually going to participate in gas exchange. Type 2 pneumocytes are the ones that produce a substance called surfactant. They produce the substance called surfactant. Now, what is surfactant? Surfactant is this fluid. It's actually a combination of lipid and a protein fluid that goes and lines the alveoli. Okay? And the purpose of that is it reduces surface tension. So the function of surfactant is to line the alveoli and decrease surface tension. Why is that important? Why must we decrease surface tension? To ensure that the lungs don't collapse during exhale, or that alveoli do not collapse during exhale. Okay? So imagine a balloon. Imagine an inflated balloon, okay? And the balloon represents basically an alveoli. The fluid, the surfactant, would be coating the inside of the balloon. So when you actually, release the balloon, when air is coming out, it doesn't completely collapse on itself. It basically still keeps it functioning. Now, where is surfactant produced? Surfactant starts being produced in our lungs at 24 weeks of gestation. Okay? That's when our lungs start producing surfactant. But it does not line the alveoli until childbirth. Because in fetal life, the lungs are not functional. So there is no surfactant that's lining the alveoli. Okay? So the production starts at 24 weeks of gestation. And then when the baby is being delivered, okay, they're going through immense stress, okay, being pushed through the birth canal, cortisol is released. Cortisol in turn causes surfactant to be secreted and line the alveoli. And after that, the baby can take the first birth. Okay? Any questions? So, example, you might have heard of a condition called hyaline membrane disease, okay, or infant respiratory distress syndrome. This is when the baby is actually born prematurely and they don't have surfactant, and it can cause a problem because they have difficulty breathing. Now let's talk about respiration. What is respiration? Movement of gases between the atmosphere and the body cells. There are two types of respiration. There's internal respiration and external respiration. Now, always remember, everything in nature always goes from high to low, meaning air flows due to the pressure gradient. It always goes from an area of high concentration into an area of low concentration. Now, what is internal respiration versus what is external respiration? Internal respiration is the gas exchange occurring between our tissues and capillaries. So, meaning between the alveoli and the red blood cell and the red blood cell and the tissue, that's internal respiration. External respiration, on the other end, is actually getting the air from the atmosphere into the lungs and back out into the atmosphere. So external is movement of air in and out of the lungs. External respiration is also called ventilation. And there are two parts, inhale and exhale. Now, air is all around us, and it actually pushes on us, presses on us. And this is why there's a specific pressure that we say. So, at sea level, the atmospheric pressure is 760 millimeters per mercury. Okay? At sea level, it is 760 millimeters per mercury. That is the atmospheric pressure. That much pressure is being exerted on our body. Now, Boyle's law states that pressure and volume are inversely proportional. If one increases, the other decreases. Okay? So they're opposite. Few vocabulary. Intrapulmonary pressure means pressure within the lungs, within the pulmonary system. And intrapulmonary volume means volume inside the lungs. Now, when we are at rest, okay, meaning you're neither inhaling, neither exhaling, there is no movement of air going on. So during rest, air is not leaving our lungs, air is not entering our lungs. Why? Because the pressure is the same. So my left hand is atmosphere, my right hand is, let's say, the lungs. The pressure outside the lungs, or the pressure in the atmosphere, and the pressure inside the lungs is both equal. It's 760 millimeters per mercury. Atmosphere is 760, the lungs are at 760. There's no movement of air. Why? Why is there no movement of air at the moment? Huh?
Pressure equalization?
Exactly, the pressures are equal. What did I say? When does air move? When there's a gradient difference, okay? When one is high, where the other is low. And right now, they're both equal.
Bless you.
Blood levels at rest. Oxygen levels are low, carbon dioxide is high. Lung volume at rest is around 400 milliliters of air. Now, how do we actually make air leave the atmosphere, come into our lungs? So if you look at my hands, atmosphere and the lungs, okay? We want air to leave the atmosphere, enter our lungs. How do we do that? Can we change the pressure in the atmosphere? No matter, you can whine, cry, get angry, it doesn't matter. You cannot change the pressure around us. But can you change the pressure inside the lungs? Yes. So you have to drop the pressure. So this is at 760, the lungs have to go down. When the lung pressure drops, there is a difference in air can enter the lungs. Okay? So how do you think, how do you propose we're going to change the pressure inside of the lungs? Any ideas? I want to cause the pressure inside my lungs to decrease. How would I go about doing that? I would increase the intrapulmonary volume. Remember I said that volume and pressure are inversely proportional. So if I want to decrease the pressure in my lungs, I must increase the volume inside my lungs. Okay? Does that make sense? Yes? So, this is how we inhale. Okay? It's basically memorization. Okay? I kind of broke it down into steps, a flow chart.
Where's one?
Excuse me?
Step one.
Okay. So, at rest, blood levels are low in oxygen, high in carbon dioxide. When your blood is low in oxygen but high in carbon dioxide, that tends to stimulate your chemoreceptors. When your chemoreceptors get stimulated, it stimulates your medulla oblongata. Why the medulla oblongata? Because that's where the respiratory center is located. So once you stimulate your respiratory center within the medulla oblongata, you tend to stimulate the phrenic nerve. That is the nerve that is reserved for respiration, the phrenic nerve. So once you stimulate your phrenic nerve, you tend to stimulate your diaphragm and respiratory muscles because your phrenic nerve innervates diaphragm and the respiratory muscle. I'm not sure if you guys recall from anatomy, but diaphragm is said to be a respiratory muscle. Okay? So now when you stimulate your diaphragm and your respiratory muscles, the diaphragm moves down and the respiratory muscles move outward. This because the lung and the diaphragm are connected. What do you think happens to your lungs when the diaphragm moves down? The lung is pulled downward. What do you think happens to your lungs when the respiratory muscles move outward? It's pulled out. So you're basically stretching your lung. Okay? And when you stretch your lung, you tend to create a negative pressure inside the lungs. So when you pull your lungs down because of moving of the diaphragm, and when you pull your lung outward, okay, they're stretched. That creates a negative pressure within the lungs. Now, a negative pressure acts like a vacuum. Let's say yesterday you were studying for your physiology midterm exam, and you had all of your handouts all laid out. Okay, the bunch of emails that Mark sent me, he printed all of that out and he said, it's all around. And now, Mark's girlfriend, wife, daughter, son, they turn on the vacuum. What do you think happens to his papers? They get sucked in. What do you think that's going to happen? I'm going to get 20 more emails from him, right? Okay, so it acts like a vacuum, meaning the lung is going to act like a vacuum and it's going to cause all of the thoracic veins to drain blood. So it's going to suck all of the blood in. Okay? So now, if the lung is sucking all of the blood in, didn't you just increase the intrapulmonary volume? Okay, so the volume inside the lungs increase. If you increase the intrapulmonary volume, what happens to the intrapulmonary pressure? It decreases. So by increasing the intrapulmonary volume, you just decrease the intrapulmonary pressure. So it went from 760 to 756. So the atmosphere is at 760. The lungs are at 756. It's only 4 millimeters per mercury difference, but that is enough to cause air to leave the atmosphere and enter the lungs. And that is inhale. Any questions? Do you need me to explain this again? No? You guys are good? Now, since we kind of touched base on chemoreceptors, let's talk more about chemoreceptors. There are two types of chemoreceptors, type 1 and type 2. Type 1 are also called central chemoreceptors. Type 2 are called peripheral chemoreceptors. Now, type 1 chemoreceptors are located near the medulla oblongata, so basically near the respiratory center. And their job is to detect hydrogen concentration within the CSF. Okay? As soon as they detect that the hydrogen concentration within the CSF has increased, that tells them indirectly that CO2 levels have increased, and they stimulate the respiratory center. Okay? So central chemoreceptors found near the respiratory center, near the medulla oblongata, they actually detect hydrogen concentration within the CSF. If the hydrogen concentration increases, it means that the CO2 levels have increased, and therefore they will stimulate the respiratory center. Now let's talk about the peripheral chemoreceptors. Peripheral chemoreceptors detect a drop in oxygen levels. Where are the peripheral chemoreceptors located? They're located in the carotid artery and the aortic bodies. Their job is to detect oxygen levels. Anytime the oxygen levels drop, it stimulates the respiratory center. Okay? Questions, concerns so far? So what if the peripheral chemoreceptors and the central are giving two different signals to the respiratory center? Let's say Mary over here is central chemoreceptors, and let's say Teresa is basically peripheral chemoreceptor. Mary is telling L, okay, he's the respiratory center, that hydrogen concentration has increased, please get stimulated. But Teresa right here is saying, no, oxygen levels are normal. You don't need to get stimulated. Which one do you think L is going to listen to, Mary or Teresa?
Whichever one was the central
Mary.
receptor.
She's a central chemoreceptor. So your central chemoreceptors is the boss between the two. So central chemoreceptors can override what the peripheral chemoreceptors are saying. Okay? Now, Mary's telling Al, okay, you know what? Fire. Teresa is also telling him fire. But I tell Al, okay, no, you're going to sit where you are. Who do you think Al's going to listen to? Me, right? So, I am higher brain center. Higher brain center can override what the chemoreceptors are telling the respiratory center. All of you guys right now, breathe fast, increase your frequency. Breathe very slow, right? You guys can do that. You started increasing or breathing very fast. Did that mean there was something wrong in your body? No. Why? Because your higher brain centers basically gave that order. So your higher brain centers can override what your chemoreceptors are telling the respiratory center. Okay? Good? Questions, concerns? This slide kind of just shows you about the chemoreceptors. I already discussed it, just so you mentioned. Now let's talk about exhale, expiration. So, due to inhalation, our lungs basically filled up with air, right? So that in turn stimulates the stretch receptors. Once the stretch receptors get stimulated, okay, they actually inhibit the phrenic nerve from working. If the phrenic nerve is not stimulated, it inhibits the diaphragm and the respiratory muscles in turn. Okay. Now, if the diaphragm and the respiratory muscles are no longer being innervated, they tend to relax. And when they relax, they come back to their original position, meaning the diaphragm moves up, the respiratory muscles move inward. Okay? That causes the lung to recoil or fall on its own property, on its recoiling property. So it kind of falls on itself because the lungs is filled with elastic tissue, elastic fibers. So because the lung is falling on itself, blood is being squeezed out of the lung. So now you're getting rid of the blood. So by getting rid of the blood, what are you doing to the intrapulmonary volume? You're decreasing the intrapulmonary volume. If you're decreasing the intrapulmonary volume, what are you doing to the intrapulmonary pressure? Increasing it. So, and by increasing it, you cause air to leave the lungs. So, during rest, look at my arms. During rest, atmosphere and the lungs were equal. Therefore, there was no movement of air. For inhalation, you cause the intrapulmonary pressure to move down to 756. That difference in pressure caused air to leave the atmosphere and to the lungs. Now, to cause air to leave the lungs, what must we do? We must increase the intrapulmonary pressure, right? We have to increase the intrapulmonary pressure. Is this going to allow air to escape? No, because it's equal. You have to go beyond. So you, during, or at the start of exhale, you're at 756, and to actually exhale, you have to bring it up to 764 millimeters per mercury. That difference in concentration is going to cause air to leave your lungs and enter the atmosphere. And how do you cause your intrapulmonary intrapulmonary pressure to rise by decreasing the intrapulmonary volume, which you did by pushing all the blood out. That caused your intrapulmonary volume to decrease, intrapulmonary pressure to increase, air now leaves the lungs. Okay. Questions? Good? Yes. So there's always 4 millimeters per mercury difference.
So 4 to get back to the regular and then 4 to...
Yes. Now, this is basically a spirometer. What a spirometer does, it actually checks your lung volumes and your lung capacities. Yes, yes. 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 Answer is B. Yes, yes. Yes, yes. Yes, yes. Hydrogen levels. So this chart table shows you the different lung volume and the lung capacities. Please keep in mind, volumes come together giving you capacities. Yeah? So this one wave, okay, one wave is called tidal volume, TV, tidal volume. This line right here, the yellow, is called IRV, inspiratory reserve volume. Please pay attention when I'm explaining these slides. It's very important because it will be on your lecture exam as well as your lab quiz. And whenever you do your respiratory rotations, all of that. So, tidal volume, inspiratory reserve volume. This black one, all the way up, that is IC, inspiratory capacity. This is ERV, inspiratory reserve volume. So ERV, inspiratory reserve volume. This right here is residual volume, RV. Now, this line is called FRC, functional residual capacity. This line is called EC, expiratory capacity. This right here is called VC, vital capacity. And this whole thing is called TLC, total lung capacity. So now let's look at what they mean, because you have to be able to identify them, understand what they mean. So this, one wave is, so this is a normal inhale and a normal exhale. So a wave represents one normal breath, one inhale and then one exhale. Okay? So inhale,
Blessing.
exhale. So TV, tidal volume means a normal breath, an inhale and an exhale. Now, after a normal inhale, you can still force your lungs to take in more, right? You could have a deep inhale, correct? That is this line right here. So IRV, inspiratory reserve volume. So amount of air you can forcefully inhale after a normal inhale, meaning it's a deep breath in. Okay? It's extra air that you're telling your lungs to take in. Now, I'm going to jump to this for a second. Now, so you could have a normal breath. You could have a deep inhale, right? You could also have a deep exhale, correct? So after you finish a normal exhale, I could still say keep on exhaling, and you can, right? That is ERV, expiratory reserve volume. Amount of air you can forcefully exhale after a normal exhale, which is this right here. So IC is inspiratory capacity, amount of air your lungs can inhale. So what do you think your lungs can inhale? It can inhale a normal air, right? Plus that extra volume. So because you can inhale this portion plus this portion, together it's called IC. Does that make sense? Okay. So you could do a normal inhale plus you could do a deep inhale. That's a total amount of inhale you can do, right? So that's why it's called inspiratory capacity. Amount of air you can inhale. Okay. So what do you think EC, expiratory capacity, means? Amount of air you can exhale. You can exhale this normal amount, correct? Plus, you could still exhale this amount, correct? So you could exhale this amount, which is the normal tidal volume, plus this extra exhale, which is deep exhale. So together, okay, it is called EC, expiratory capacity. So do you see how the volumes come together giving you the capacity? Now, this right here is residual volume, RV, residual volume. What does residual volume mean? Amount of air always left in the lungs. You cannot touch that amount of air. Okay? So after you do a normal exhale, what air do you have left in your lungs? The residual volume, the extra that you can exhale, plus you have this one left over, right? Right? So after you finish a normal exhale, you have this amount that you could exhale, plus you have this one, right? Those two come together giving you FRC, functional residual capacity. Amount of air left in your lungs after a normal exhale. So once you're done with a normal exhale, you still have this amount of air left, plus you have this amount of air left. Together, these two is called FRC, functional residual capacity. Good? Now, this right here is VC, vital capacity. This is the amount of air that you can manipulate, you can change, you can alter it. Okay? And then, of course, can you touch this part? No. This is why vital capacity does not contain this, because you can't do anything with that amount of air. It's always there. Okay? With total lung capacity, it's everything. Total amount of air in your lungs. That is total lung capacity. Questions? It's good?
I do have a question.
Yes.
On, what plus what together equals the, or comes together with the EC?
Okay. So EC is amount of air you can exhale. You could exhale the normal tidal volume, right? Plus you could exhale ERV. So ERV and TV give you EC.
Oh, thank you.
What is this called?
Third.
Perfect. What is this called? What is this called? What is this called? Good. What is this called? EC. What is this amount of air? What is this called? What is this called? Not TLC because I'm not involved in this. So now, all of this is TLC. Does that make sense? Okay? Yes.
So FRC includes the reserve too.
Yes. So FRC is amount of air left in the lungs after you finish a normal exhale. After you finish a normal exhale, you have the ERV left over, plus you have the RV, which is always there. Okay. Good? Move on. So, this basically chart table helps you understand what volumes come together giving you capacities. So, IRV and TV come together giving you IC. ERV, RV come together giving you FRC. TV, ERV come together giving you EC. Now, VC is IRV, TV, and ERV give you VC, right? Yeah? So these three come together giving you VC. VC plus RV come together give you TLC, right? So when you add TV and ERV, that gives you EC. When you add TV and ERV, that will give you EC. Just look at the boxes right next to it. These two come together giving you the blue box. These two give together the yellow box. Yeah, VC included. VC is IRV, TV, ERV. It gives you VC.
Okay.
but not IC or EC.
Okay, the equation for VC could be IC plus ERV, or it could be EC plus IRV.
That's where you're going.
Because all three of these equal VC. If you combine these two, it gives you IC. Right? So it would be these three together give you VC. So these three are already in this one. So it's this plus this giving you VC, or it's this plus this, or this plus this giving you.
That's where we're going.
Gives you VC. Okay. Or you can just do IRV plus TV plus ERV. Because when these two come together, that gives you IC right there.
Right?
So if you add these two, that's IC, what do you get? Extra is ERV. If you add these two, that gives you VC plus this one. And VC plus RV give you TLC. You have to understand this. From every single exam from now on, you will have math in your exams. Okay? You have to remember formulas. You will have to plug in stuff and calculate the stuff. Okay? Every single exam from now on, there is going to be math. We will give you calculators. But the math is not going to be decimal points and stuff like that. It's going to be simple math. And because I'm trying to do simple math, it might not actually be the true num values, but I'm trying to see if you understand the concept. Okay? Good? Can I move on? Okay. So let's calculate this. Yes, 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 yes. Yes, yes, yes. Yes. I need more students to participate in this. I only had I have 50 students. Yes. Yes, yes, yes. Yes, yes, yes. If you don't have a board, use a paper, write it out. Yes. No more. Yes. 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 Yes.
You just didn't have a board, so I didn't go writing.
Yes, yes.
Oh, we got it highlighted, right?
Maria, I need yours. Answer. Yes. 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 Christina, I need your answer. Oh. Maddie, need your answer. Back there. Okay. Yes. Answer is 500. How?
IC.
How do you calculate IC? What is TV? What is IRV? Gives you 500. I do, but I'm not sure why I do. No. So, Maria, no. Jason, yes. No, no, yes, no, no. Yes. 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 No. Yes. No. No. Yes, ma'am.
How they got the
Yes. Yes.
table? It's gonna end up on the show taking out.
Let them try again. Let them try. Yes. So you have to figure out an equation. So you have to use two equations. So the answer is B, 900.
Take half of the F.
So what is the equation? Wyatt? How do you figure out VC? What is the equation?
TB plus IRV.
So IRV plus TB plus ERV. Correct? What is IRV?
200.
What is TB?
300.
What is ERV? How do you get 400? It's not really. So what is the equation to figure out ERV? What is the equation to figure out ERV? So to figure out ERV, it's FRC minus RV. What is FRC? What is RV? So what do you get?
Both.
So 600 minus 200 gives you 400. So 200 plus 300, 500. 500 plus 400 is 900. So you have to use two different equations. I gave you stuff directly for the first equation, but I did not give you something for the second one. You have to use a different equation.
You have to have this table memorized.
Would I say do you have to memorize a table? No. You should understand the relationship between the capacitors and the volumes. So last term a student asked me this, do I need to memorize it? And I said no. They're like, is it going to be on test? I'm like, yes. So they're like, you have to memorize it. I'm like, you have to understand it. Okay, so if you have to memorize it, you memorize it. If you can understand it, then you. Now listen up. On the test, I don't need to give you the chart. You should be able to draw the chart out. Okay, and label all the stuff. So what I've had students do is when they come across, they draw all of the chart out, they label each part, and then the values I give, they plug it in, and then they do the math. Okay? I'm not that nice to give you the chart on that exam. Okay? Okay, now, this slide, I do not need to memorize it, it's just trying to show you a concept that air always flows due to the pressure gradient, the difference. So if you see in the alveoli, we have high oxygen, but in the capillary, low oxygen. This is why oxygen leaves the alveoli, enters the capillary. Well, the capillary has more carbon dioxide than the alveoli. This is why carbon dioxide enters the alveoli. When you look at your tissues, in the tissues, the capillary has more oxygen compared to your tissues, so oxygen enters your tissues. Where your tissues have a higher level of carbon dioxide, this is why it leaves the tissues, enters the capillary. So everything is always going from high to low. Okay. Now, yes?
Can we go back and explain that one more time?
So that would just be straight up confusion, correct?
Yeah. It's just diffusion. Everything is always going from high to low. Alveoli has high oxygen. This is why it leaves the alveoli and enters the capillary, which is low levels. This is high, this is low, carbon dioxide. That's it. This is lipid diffusion. Okay, oxygen transport. So this is your alveoli. This is your capillary, and these are your red blood cells. When oxygen leaves the alveoli, 98% will enter a red blood cell and bind to hemoglobin. So, 98% of oxygen will bind to hemoglobin. And when oxygen is bound to hemoglobin, we call it oxyhemoglobin. About 2% just dissolves in plasma. So 98% is bound to hemoglobin. Roughly 2% is dissolved in plasma. That's how oxygen is transported in our blood. Now, we're going to see how carbon dioxide is transported in venous blood. Meaning, when carbon dioxide is released by our tissues, it could be our lungs, well, not lungs, it could be our heart, kidneys, spleen, eyes, etc. When carbon dioxide leaves it and enters the capillary, how is it transported in venous blood? There are 3 ways. % of CO2 will leave our tissues, enter a red blood cell. And in a red blood cell, it binds to water. So 70% of carbon dioxide enters a red blood cell, binds to water. When you combine water, carbon dioxide and water, you get carbonic acid. Carbonic acid is broken down into hydrogen and bicarbonate via an enzyme called carbonic anhydrase, which is written right here. Ca stands for carbonic anhydrase. So you form hydrogen and bicarbonate. This bicarbonate leaves the red blood cells. In exchange, chloride comes in. So the bicarbonate leaves the red blood cell and stays in the plasma in that form. And in exchange, chloride comes in. This transporter that allows bicarbonate to leave, in exchange chloride to come in, is called alkaline tide. So we say, because didn't this carbon dioxide basically turn into bicarbonate? Right? So we say 70% of carbon dioxide is transported in venous blood as bicarbonate. % of CO2 20% of CO2 leaves the tissues, enters a red blood cell, binds with hemoglobin. 20% leaves the tissue, enters a red blood cell, binds to hemoglobin. And it's transported in that form. What do you say when carbon dioxide and hemoglobin are bound? You call that molecule carboaminohemoglobin. So 20% of carbon dioxide is transported as carboaminohemoglobin. 10% of carbon dioxide leaves the tissues and enters the plasma, and it dissolves in plasma. So there are three ways where carbon dioxide is transported in venous blood. 70% as bicarbonate, 20% as carboaminohemoglobin, meaning binding to hemoglobin, and 10% just dissolves in plasma. Now, this is a capillary, so it just picked up all of the CO2. Where do you think the end destination for the CO2 is? Lungs, right? Isn't this carbon dioxide supposed to go to the lungs so we can exhale it? So what happens when this blood, which is filled with carbon dioxide, how is it releasing the CO2 now? So now this carbon dioxide rich blood has arrived at the lungs. How is it releasing the carbon dioxide? Look at the image. Can you see something, a pattern? Okay, so yes, it kind of leaves the capillary, enters the alveoli. But my question is, how is it able to leave? Look at this part of the image. What can you say? It's the opposite. It looks identical to the previous slide, right? The only difference is the arrows are backwards now, right? Now, if I go back, look at, right now, memorize this image, just kind of quick glance, okay? Now, do you see it's identical, only the reverse? So, the 70% of bicarbonate, the 70% that we had made into bicarbonate enters back in exchange, chloride leaves. That bicarbonate binds with hydrogen, giving you carbonic acid. That carbonic acid breaks down to CO2 and water, and CO2 leaves. So when we were taking it in, it went this way. Now when we're throwing it out, it goes this way. The 10% of CO2 that had dissolved in plasma just leaves. That 20% that we had transported as carboaminohemoglobin, now it detaches from it detaches from bicarbonate. It detaches from hemoglobin and it just leaves. So the cycle is complete opposite. Yeah? Good? Okay. Mark, can you close it for 5 minutes? Take a 5-minute break. Because the next part becomes a little bit complicated. All right, listen up. So now what we're going to cover is acid-base disturbance.
That's actually pretty good though, like on this slide, you do that.
Before we cover acid-base disturbance, let's kind of review about pH. So pH is a concentration of hydrogens, free hydrogens in a solution. pH ranges from 0 to 14. 0 to 7 is acidic. 7 to 8 is neutral. 8 and above is basic, or we call it alkaline. Now, acid-base disorders. There are two main classes of acid-base disorders. You have acidosis or alkalosis. Acidosis means that you are acidic. Your pH is low. Versus alkalosis means that your pH is alkaline or high, okay, meaning you're basic. Now, there are only two main types of acidosis or alkalosis. So you could have metabolic acidosis or respiratory acidosis. So you can have metabolic acidosis or respiratory acidosis, or metabolic alkalosis or respiratory alkalosis. So there are only two main types, metabolic or respiratory. When we say respiratory, respiratory deals with CO2 because CO2 is acid, okay? So when we say we have respiratory acidosis, it means we have too much CO2 because CO2 is acid and you are acidosis, okay? So respiratory acidosis means you have too much CO2. Respiratory alkalosis means that you don't have enough CO2. And because you don't have enough acid, which is CO2, you are alkaline. Okay? Now, respiratory means CO2, meaning it deals with the respiratory system. Your respiratory system is at fault. When you call something respiratory acidosis, respiratory alkalosis, you're saying that your respiratory system is at fault and screwing up your CO2 levels, basically. Okay? If there's any other reason for you to become acidic or for you to become alkaline, but it has nothing to do with your respiratory system, then we call it metabolic. It could be you drank too much acid. Let's say you drank a lot of orange juice, which is acid. Your pH becomes acidic, meaning acidosis. Is it your respiratory system's fault? No, so it's metabolic acidosis. Okay? Or you don't have enough base, which is bicarbonate. Let's say you have gotten rid of all of your base. So now in your body you have too much acid, right? Causing you to go in acidosis. Is your respiratory system at fault? No. So you call it metabolic alkalosis. Okay? Now, when you say you have metabolic alkalosis, it means that you have too much base. Because you have too much base, you call it metabolic alkalosis. Or it could be you don't have enough acid in your body. Because you don't have enough acid in your body, you're in alkalosis. It has nothing to do with your respiratory system, therefore it's called metabolic alkalosis. Does that make sense? Now, compensation. Compensation is basically a way to bring your pH back towards normal. So your body compensates anytime there's a disturbance, anytime you become acidic or alkaline. Your pH will try to come back to normal, and that is called compensation. Your opposite system compensates. So if your respiratory system is at fault, your metabolic system compensates. If your metabolic system is at fault, your respiratory system compensates. Of course, respiratory system compensation is much faster than your metabolic. It may take time to actually see the metabolic compensation occurring. So, if you have metabolic acidosis, your respiratory system will compensate. How? Your respiratory system will compensate by hyperventilation. So when you hyperventilate, you get rid of your CO2, bringing your pH back down. When you say you have respiratory acidosis, your metabolic system will compensate. How would your metabolic system compensate in respiratory acidosis? It will retain the bicarbonate, so it will increase bicarbonate reabsorption from the kidneys. Okay? So in terms of compensation, your metabolic is your kidneys, think of that. So when you are respiratory acidosis, your kidneys will reabsorb the bicarbonate, trying to bring your pH back up. When you say metabolic alkalosis, your respiratory system will compensate. How? It will hypoventilate because you don't have enough acid. So it will retain the CO2. CO2 is acid. So your pH comes back up. I mean, not back up, sorry, goes back down. When you say you have respiratory alkalosis, your metabolic system, which is your kidneys in terms of compensation, will compensate. How will your kidneys compensate? It will increase the secretion of bicarbonate. So you're getting rid of the bicarbonate in your urine. By getting rid of the bicarbonate, your pH starts going back down. Yeah? Does that make sense? Okay. Now, you should be able to look at a person's blood values, blood gases, and say if there are a disturbance, and if they are, what type of disturbance. So you look at three things: pH, CO2, and bicarbonate. And from there, you could tell what type of acid-base disorder that person has, and then figure out what the compensation would be. Okay? So I've given you the normal values. I have not informed you what type of acid-base disorder that person has. That's your job to figure it out. So I will help you figure out the first one. We'll work together as a class. Then you're going to have about 2-3 minutes to figure out what type of acid-base disorder the rest of the values are, and what type of compensation would occur. Okay? Any questions? Okay. So, the first thing you do is you look at the pH, and you figure out what the pH is. So 6.9, is that increased or decreased? So how do you call it when the pH is decreased? Acidosis. So you know that you're in acidosis. Either it's metabolic acidosis or respiratory acidosis. There are only two types. So now you have to figure out which one. Second step is you look at the CO2 levels. Okay. So the first step is you think, if you have respiratory acidosis, okay? If somebody has respiratory acidosis, what would the CO2 levels be?
High.
Right? If somebody has respiratory acidosis, the CO2 levels should be high, correct? Okay. So you think that it's increased, correct? That it should be increased. Then you actually look at the value. Is it increased or decreased?
Decreased.
Okay. So does it match your prediction? No. If it does not, so it should increase, but it has decreased, then you know it cannot be your respiratory system. So it's your metabolic. So if it does not match your prediction, it's the opposite system at fault. So third step is you look at the bicarbonate, just to reconfirm your answer to see if it's right. So then you think, if somebody has metabolic acidosis, what should the bicarbonate levels be? Because that is your base. If you have metabolic acidosis,
No, it's low.
your bicarbonate should be low. Now you look at the value. Has it decreased?
Yep.
Does it match the prediction? If it matches the prediction, then you call it that. So this person has metabolic acidosis. Okay? And then the fourth step is compensation. So now you know it is metabolic acidosis. Which system will compensate? Respiratory. What will your respiratory system do? Hyperventilate. Why? Because if you hyperventilate, you're getting rid of your CO2, which is your acid, bringing your pH back up. So the CO2 levels are decreased in blood, okay, by hyperventilating. So you're getting rid of it. Now, take about 2 to 5 minutes to figure out what type of acid-base disorder these are, and then what the compensation would be. So that's your job now. When you say incarceration, it means it gets rid of it. No, reabsorption means you're taking it out. Secretion means you're getting rid of it. Yes.
It's really good YouTube.
Anyways, well, I think that one was testing.
Yeah, that's why I said YouTube.
It's like 20 minutes or less. This right here.
Metabolic alkalosis with the compensation as well. Yes. You got, what do you put? Because the respiratory is going to be respiratory alkalosis, but not enough CO2. But his CO2 level is way too high. So it's not going to be respiratory alkalosis.
I'll let you figure out if your arrows are the same direction.
It's not respiratory, it's CO2.
If they're counterintuitive,
Yeah, the second one's metabolic. So what if you don't? For the second one, because like whatever's wrong in the constantly gets compensated by the so it's metabolic. It's already high, that's what I was looking at too.
Okay, so let's figure out what type of acid-base disorders the next ones are. So, 7.5, is that increased or decreased? So if the pH is increased, you call it? So now, either respiratory alkalosis or metabolic. If you have respiratory alkalosis, what should your CO2 levels be? It should be low. Is it low?
No.
No. So then it is called metabolic. If you say you have metabolic alkalosis, what should your bicarbonate levels be? High. Has it increased? Yes, it's increased. So you know this is right, metabolic alkalosis. So which system compensates? What will your respiratory system do? Hypoventilate. Why? To increase CO2 levels. Okay. Now let's look at the third one. Does everybody understand this? 7.1, is that increased or decreased? It's acidosis. If you say you have respiratory acidosis, what should your CO2 levels be? Increased. Has it increased? Yes, so it is. Which system compensates? So metabolic system. Or metabolic system. Which, in terms of compensation, when you say metabolic, it means kidneys. but you say metabolic. So what will your metabolic system do? What will your kidneys do? It will increase reabsorption of bicarbonate. It will increase bicarbonate reabsorption in the kidneys. So your kidneys will increase reabsorption of bicarbonate. Last one. 7.6. Is it acidosis or alkalosis? If you say you have respiratory alkalosis, what should your CO2 levels be?
Is it respiratory alkalosis?
Whoa.
It should be low. Is it low? Yes, it is low. So it is respiratory alkalosis. What's the compensation? Metabolic system. What will your metabolic system do? It will increase bicarbonate secretion in the urine. Okay? So when you say reabsorption, it means it takes it back. When you say secretion, it means it gets rid of it. Yeah? Good? Answers on the next slide.
I feel like once I go over it once.
So these are the answers for you.
I know she's not going to use that
Yes.
bag.
Yes?
I know you
Yes.
don't even want to do it either.
No.
She'll appreciate that.
Yes. Answer is D, metabolic acidosis. Why? It tells you phosphoric acid is being produced. Acid throws you into acidosis. Is it your respiratory system's fault? No. So it's called metabolic acidosis. No. I didn't see yours. No. Yes, Marie. Yes. Yes, yes. No. No. Yes. Yes, Nathaniel. No. No, Mark. No. Yes, yes. No.
I think it's metabolic.
Yes, Christian. Monica? No.
Yeah.
No. Yes. No. Respiratory alkalosis. It tells you the female starts hyperventilating. If you hyperventilate, what happens to your CO2 levels? They decrease. So it throws you into alkalosis. And this is your respiratory system's fault, right? So it's called respiratory alkalosis. We're done with the respiratory system. Mark, can you please close the recording? Okay, so this is our second lecture for today, which is the muscular system. Okay. CLO, objective, explain the structure of thick and thin filaments, explain how muscle contraction occurs in skeletal muscle via neuromuscular junction, excitation, contraction, coupling, and sliding filament theory, as well as explain how contraction occurs in smooth muscle. So, when you look at a muscle cell is just filled with structures called myofibrils. What is the importance of myofibrils? Myofibrils are just filled with sarcomeres. Here's one sarcomere, here's another sarcomere, here's a third sarcomere. Sarcomeres contain contractile proteins. Now, all of the myofibrils are surrounded by this membrane. In the image, the membrane is blue. This membrane is called sarcoplasmic reticulum. It contains calcium. So sarcoplasmic reticulum is a calcium storage site. Apart from that, you see this yellow area. This yellow area are transverse tubules or T tubules. Think of transverse T tubules as a hallway or passageway. It allows the action potential to travel from the outside of the cell to arrive interior of the cell. Now, taking a closer look at the T tubules and the sarcoplasmic reticulum. Now, this is the muscle cell. Okay? This is the cytoplasm of the muscle cell. This is the plasma membrane, and this is the T tubules. Here is the sarcoplasmic reticulum. Now, the T tubules, at the end of it, has the specialized receptor, the protein. The name of this protein is called dihydropyridine receptor. Okay? So, transverse tubules has the specialized protein at the end, and that is called dihydropyridine receptor. You should know that name. SR, the sarcoplasmic reticulum, contains calcium. So all of these blue circles are calcium. Sarcoplasmic reticulum has this receptor right here, which is called rhinodine receptor. So the name of the receptor that belongs to the sarcoplasmic reticulum is called the rhinodine receptor.
Oh, the gray one.
Yes, the green one. So the way the sarcoplasmic reticulum opens up, okay, it's controlled. It's calcium transport is controlled by the rhinodine receptor. So this is the rhinodine receptor. Okay, imagine I'm holding a balloon, and the balloon is inflated. Okay, so I'm holding it right now, closed tightly. Is the air able to escape? And imagine air as calcium. Is air able to escape? No. But what happens if I go like this? Air is going to escape. Same concept. So this is the rhinodine receptor. When the rhinodine receptor is closed, calcium is not able to escape. When it opens up, calcium is able to escape the SR. Okay? Now, that is the rhinodine receptor. Now let's talk about the dihydropyridine receptor, which is part of the transverse tubules. So imagine my arm as a transverse tubules, and my palm, okay, my hand itself, becomes the dihydropyridine receptor. The two proteins are connected. So the dihydropyridine receptor and the rhinodine receptor are connected to each other. So this is how they are. Imagine I have a balloon, okay? So when this dihydropyridine receptor, when it basically gets activated or depolarized, it basically opens up. It kind of blossoms like a flower. And because they're connected, when one opens, it causes the other to open up. Does that make sense? So when the rhinodine receptor, okay, sorry, not rhinodine, when the dihydropyridine receptor, when it opens up, it actually causes the rhinodine receptor to open up. Okay?
Does it go vice versa?
No.
Just that opens first.
Exactly. So it's always the dihydropyridine receptor which has to depolarize, which causes this to blossom or open up, and then that in turn causes the rhinodine receptor to open up, allowing calcium to escape. Keep that in mind. It comes into play towards the other half of the lecture. Now, this SR, apart from this receptor, it has a transport protein right here. This is called the calcium ATPase pump. This is how the calcium is put back into the SR. Okay. This is the sarcomere. One sarcomere extends from one Z line to the second Z line. Okay? Sorry, Z line. One Z line to the second Z line. This is the length of a sarcomere. Right in the middle, we have an M line. This is where you find the thick and thin filaments in the sarcomere. Going from the M line to the Z line, you find thick filaments. Going from the Z line towards the M line, you find thin filaments. Okay? You can see the thick filaments are indirectly attached to the Z line, right? Via this squiggly blue lines. That is a protein called titin. Okay? So a protein called titin connects the thick filaments to the sarcomere. Not to the sarcomere, to the Z line. Now, apart from the lines, they actually have zones and bands. This area right here is the H zone. And what filament do you think, by looking at the image, what filament is located underneath the H zone, or within the H zone? Okay. Only thick filaments. Right here is the A band. Which filament do you find under the A band? Only thin? And thick and thin. So A band is the part of the sarcomere where the thick and thin filaments overlap each other. And then the I band. What filaments do you find in the I band? Only thin. It only contains the thin filaments. I band only contains the thin filaments.
H is thick.
H is thick, I is thin, A is both. Now, here is the length of a sarcomere when the muscle is relaxed. When the muscle contracts, you can see the length of the sarcomere shortens. What disappears, the I band is gone. A band remains the same length. The I band disappears during contraction, okay? But the A band remains the same length. What allows it to go from a relaxed state to a contracted state? ATP and calcium. Now let's look at thin filaments. Thin filaments are made up of a protein called actin. So these are actin molecules. Each actin molecule has a specific site. This is called a myosin binding site. So active site or myosin binding site. Because that's where myosin will actually attach. All of these proteins, these molecules, come together creating chains. So thin filaments is made up of two chains, two actin chains. These actin chains are actually regulated or overlapped with another structure called TTC, troponin tropomyosin complex. So the yellows is the troponin tropomyosin complex. Okay. So what is troponin tropomyosin complex? Troponin is a protein. Tropomyosin is another protein. Both of them are found together, attached. This is why it's called a complex. Tropomyosin is the green line. It's basically a fibrous protein that looks like a rope. Troponin is a protein which is circular, like globular, okay? And it has three different subunits. It has TC, TI, TT. Okay? TT stands for tropomyosin, the I stands for interaction, and the C stands for calcium. Okay?
Is that tropomyosin or troponin?
Well, the first T's are troponin. So it's troponin, calcium, troponin interaction, troponin, tropomyosin. Now, the calcium one. The TC component has a calcium binding site on it. This is where calcium will bind. The TC component allows calcium to bind. So what do you think the function of the TTC is, the troponin tropomyosin complex? Its function is to cover up the myosin binding sites on actin. So the blues are the actin chains. And you can see the way that the TTC is overlapping it, it's closing up the myosin binding sites, right? So thick and thin filaments cannot interact. So think of this chair as basically actin molecules, okay? And the space where you could actually sit is the myosin binding sites. This is where myosin will bind. Quinn, can you come up here? So Quinn is myosin, okay? The chair is actin, okay? And where you could actually sit on the chair is that myosin binding sites. And my body is tropomyosin, my arm is troponin, and my palm is the last component, the TC component of troponin, okay? So right now, okay, can Quinn sit in the chair? No, why? I'm blocking it. So as soon as I basically move out, as soon as the TTC moves away, myosin can actually combine, okay? So what allows me to move away? So if I'm right here, what allows me to actually move away? Calcium, can you be a marker? So this marker represents calcium. As soon as calcium comes and binds to a TC component, it kind of pulls the rest of the protein out. Therefore, this is exposed. Thick and thin filaments can actually combine, which is called cross-bridge formation. Okay?
Whenever calcium binds, I'm automatically going to lose evolution.
Yes, yes.
So it's about finding.
Actin and myosin always want to be attached. But as long as tropomyosin is there, it cannot. As soon as tropomyosin moves away, it's like I can finally go sit down. I'll go down. Okay. Now let's talk about the thick filaments. Thick filaments are made up of molecules called myosin. Each myosin has a tail and two heads. The heads are the one that actually binds to actin, okay? And each head actually has an ATP binding site, meaning it allows ATP to bind to it. When you look at thick filaments, all of the tails are in the middle. Protruding outwards are the heads. And each head would have an ATP bound to it. Now, ATP and myosin. There's two ways ATP can bind to myosin. You could have ATP bound as ATP, okay, pure form ATP, which in that state we say that myosin is in low energy configuration. Or ATP can be bound as ADP and PI. When it's bound in this way, we say myosin is in high energy configuration. Doesn't that sound weird? Right? Why? Why do you think it's that way? Why is it that ATP is considered low energy, where ADP and PI is considered high energy? Well, yes, kind of. Okay, imagine, let's say, Danielle, I give you $100. Okay? Now, at 2 o'clock in the morning, let's say you're starving, you go to Taco Bell. Are you going to be able to buy anything with that $100 bill? No. They're not going to accept a $100 bill. That's, okay, but now, assume I give you $100, but I give you in tens. So I give you 10 tens. Now at 2 o'clock in the morning, you go to Taco Bell. Are you able to buy food? Yes. Why? Because they will not accept the $100 bill, but they will accept tens. So that's the same concept. This is saying it's a $100 bill, where myosin cannot use it. Where this is saying it's $100, but in forms of tens. And this is why it's called high energy configuration, because myosin can actually use the energy. Okay? So this is saying that it's in bill, where this is $100, but in tens. Okay? Yes?
So you're saying since it's broken down, the ADP and PI, that's why it's usable, whereas?
Yeah.
Okay.
Yeah. It's because each component, the phosphate and the ADP, cause myosin to do two different things. That's why. Okay, so this is the state when there's relaxed. You can see there's no interaction between thick and thin filaments. This is during contracted state. You can see there's interaction between thick and thin filaments. Special note. Yes. No. Yes. Okay. So it is D. But which one is F4? Dihydrogen receptor. Now, what structure is it? The dihydrogen receptor is F4. It's saying, it's asking, do you know that dihydrogen receptor is part of it? Okay. Listen up, you guys. We have half an hour. Okay. Now, understanding the muscle contraction. 4 things. Skeletal muscle has to be innervated by a nerve. When it's innervated, calcium levels increase around the contractile proteins. Calcium permits contraction. Absence of calcium prevents contraction. When the nerve is no longer stimulating or innervating the muscle relaxes. Okay. Now, this is muscle contraction in skeletal muscle. In skeletal muscle, there are 3 processes involved in skeletal muscle contraction. The first one is neuromuscular junction process. Second one is excitation contraction coupling. Third one is basically sliding filament theory. Okay. So neuromuscular junction process. This is the point where the neuron releases the neurotransmitter and it binds to the muscle cell. That's neuromuscular junction process. Excitation contraction coupling is where calcium is released and cross-bridge formation occurs. Sliding filament theory is the step where actual contraction occurs. They go in this order. Okay. Now, the neuromuscular junction process is the same as synaptic transmission. Okay? So, these are the steps of synaptic transmission. They're not in order. I usually would give you time to kind of place them in order, but because we're running out of time, the correct order is on the next slide. So neuromuscular junction is identical to synaptic transmission. Okay. 2 things that I want to clarify about neuromuscular junction, which is a little bit unique to it, or specific to it. Acetylcholine is the neurotransmitter in the neuromuscular junction. Okay? You cannot have any other type of neurotransmitter in a neuromuscular junction. It's always going to be acetylcholine. Now, once acetylcholine is released, it's going to bind to a receptor on the muscle cell, right? And of course, it will always open up a channel. So, this is the muscle cell. Okay? This is a muscle cell, and right here is the protein. And the red is the receptor. So the red is the receptor to which acetylcholine binds, and that receptor is actually a ligand-gated channel. So what is this ligand-gated channel? It's a ligand-gated sodium-potassium channel. Okay? And what is the name of the receptor? It's nicotinic cholinergic receptor. So name of the receptor is nicotinic cholinergic receptor. And the protein is ligand-gated sodium-potassium channel. Okay. So as soon as a neurotransmitter is released and binds to its receptor, we're done with neuromuscular junction process. Now you go into excitation, contraction, coupling. Okay, in the excitation, contraction, coupling, what I have done, it's basically one big photo. I've cut it up into two, so we can see the process more directly. So, as soon as acetylcholine binds to its receptor, opening up ligand-gated sodium-potassium channel, sodium comes into the cell, potassium leaves. But there's more sodium coming in. So what do you think happens to the membrane potential due to entry of sodium? It becomes positive, reaching threshold. What happens once you reach threshold? Action potential occurs, right? Do you guys remember the nervous system?
Yes.
As soon as you reach threshold, action potential occurs. So the net entry of sodium causes the postsynaptic membrane to depolarize or reach threshold, causing action potential to occur. Reaching threshold causes action potential to be generated. So right here, you see the action potential is generated on the surface. Now the action potential travels down the T tubules. What was at the end of the T tubules? The dihydropyridine receptor. So the action potential comes down the T tubules, depolarizing the dihydropyridine receptor. What is the dihydropyridine receptor connected to? Ryanodine receptor, causing the ryanodine receptor to open up. Once the ryanodine receptor opens up, what do you release? Calcium. So action potential travels down the T tubules, causing the dihydropyridine receptor to depolarize, causing the ryanodine receptor to open up, calcium escapes. Okay? So that is this part of the image. We just released all of this calcium, and now you can see calcium is going into the sarcoplasmic reticulum. Whatever. Now, calcium arrives at the sarcoplasmic reticulum. Where do you think it's going to go bind?
To the TC.
The TC component.
So it can move it.
So right here in the sarcomere, you see thick and thin filaments are not connected. Why? Because the myosin binding sites are closed. Now calcium comes, binds to the TC component, causing TTC to move over, exposing the myosin binding sites. As soon as they're exposed, thick and thin filaments connect to each other, which is called cross-bridge formation has occurred. As soon as cross-bridge formation occurs, we are done with excitation, contraction, coupling. And we move on to the last part, which is sliding filament theory. Tina, this is where now your thought process comes in. No. Yes. 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 No. Yes. So the answer is C. The ryanodine receptor will not open up. Sliding filament theory, which is this one. let's do the sliding filament theory. This is where, listen up you guys. Sliding filament theory is where you actually see the contraction going on. So, at the end of excitation contraction coupling, this is where we are. We just caused thick and thin filaments to combine. The first step in excitation contraction the first step in sliding filament theory is to release ADP and PI. So ADP and PI are released, causing the myosin head to bend. When the myosin head bends, thick and thin filaments slide past each other, which is called the power stroke, or the working stroke. Okay? So ADP and PI are released, causing the myosin head to bend. Thick and thin filaments slide past each other. That's actually the contraction. That is the working stroke or the power stroke. Now, a new molecule of ATP comes in. As soon as ATP comes in and binds to myosin, it causes thick and thin filaments to detach. But the head is still bent. Now, the ATP is converted into ADP and PI, the high energy state. That causes the head to straighten up again. So, hydrolysis of ATP causes the myosin head to straighten up. Once the myosin head straightens up, what happens? So, once you come back to this state, either it goes root A and the cycle happens again, or it takes root B, which muscle relaxes. What will determine if root A occurs or root B?
The calcium.
Calcium. Okay? So if calcium is present, the cycle happens again. But if there's no calcium, muscle relaxes. But what will determine if calcium is there? The action potential. What will determine if the muscle cell is generating the action potential? What will determine if the muscle is generating the action potential? If acetylcholine is basically released and binding to its receptor. What will determine if acetylcholine is being released? Basically, is the nerve innervating the muscle? If the nerve is innervating the muscle or stimulating the muscle, calcium is present. If the nerve is no longer stimulating the muscle, okay, there is no calcium being released. Therefore, the muscle relaxes. Okay? Okay. All right. Okay. Look at me for a second. This is ADP and PI. Okay. Myosin tail, myosin head. This is thin filaments. This is the myosin binding site. And my thumb acts like a TTC. As soon as calcium is bound, that won't stay on. Okay. That won't stay on either. Okay. Imagine I have calcium. So as soon as calcium is present, the myosin binding sites are open, correct? So thick and thin filaments interact. This is, at this point, you start the sliding filament theory. The first step in sliding filament theory is a release of ADP and PI. As soon as ADP and PI are released, myosin head bends, causing the thick and thin filaments to slide past each other. As soon as that happens, this is called the power stroke. Now a new molecule of ATP will come and bind to it. Take the yellow marker. So this is ATP. When ATP binds to myosin, it causes the thick and thin filaments to separate. Okay? But you can see the head is still bent. As soon as this ATP molecule is converted back into ADP and PI, it causes the head to straighten up. Okay? If this is still exposed, the cycle happens again. Okay? Does that make sense? But by the time it gets converted back into ADP and PI, if this is closed, okay, by the time it straightens up, if this is closed, it cannot bind. So there's relaxation going on. Okay? Does that make sense? Question, concerns? So this is the sliding filament theory. So this is another photo just showing you sliding filament theory. Some students prefer this photo, some students prefer the previous one, whichever one you like. Okay. Now, this is just a summary of muscle contraction. It's summary from neuromuscular junction process till sliding filament theory. Just not in details, just a good overview. This slide, this slide. Okay? Question 3. Good. Yes. 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 Okay. So the answer is E. Myosin head doesn't straighten up. Okay. This is how the calcium is put back. So the calcium we had released, how is it put back? By the calcium ATP is pumped. It's actively put back in. Okay. These are basically hyperlinks to basically YouTube videos that show neuromuscular junction, excitation, contraction, coupling, sliding filament theory. I highly tell you to watch these. These are extremely good. Okay? And I know if these links don't work, your mastering homework that I assigned this week for lab basically, it also contains these videos because these videos are on YouTube but they're Pearson product. So you have the same videos in your mastering homework. Okay. The last thing I need to teach you is smooth muscle contraction. It's a little bit simpler than skeletal muscle. Okay. So, before I explain the smooth muscle contraction, I need to kind of lay some foundation. Smooth muscle contains this protein. It's called calmodulin. This protein is inactive. Okay? So smooth muscle contains a protein called calmodulin and it's inactive. Smooth muscle also contains another inactive protein which is called myosin light chain kinase. The name is right here. It's basically an enzyme that is inactive. Okay? Smooth muscle also contains myosin, but the myosin is also inactive. Why? It does not have any energy. Okay? So smooth muscle has 3 inactive proteins: calmodulin, myosin light chain kinase, and myosin. To cause the thin filaments to interact and actual muscle contraction to occur, you actually have to activate these 3 proteins. Only when these 3 proteins are activated is there muscle contraction in smooth muscle. So we're going to see how these 3 proteins are activated and how muscle contraction occurs. So what I have done, I've taken this image, cut it up into 2. So we can just see it better. So here is the muscle cell. This is smooth muscle cell. This is the plasma membrane. On the smooth muscle plasma membrane, we have 2 types of calcium channels. It's only shown 1, but it's kind of says there's 2 types. There is so there are 2 types. There's voltage, so voltage gated calcium channel or voltage independent calcium channel. Okay? So there are 2 types. There's voltage dependent calcium channel and voltage independent calcium channel. So calcium comes in via the voltage dependent, voltage independent calcium channel. As soon as this calcium comes in, this calcium actually goes, so that's step one, entry of calcium. As soon as that calcium comes in, it actually goes to the sarcoplasmic reticulum, causing it to open up, releasing more calcium. Okay? So the calcium from the extracellular fluid comes in, causing the SR, the ryanodine receptor, to open up, allowing more calcium to come out. And you can see calcium is released from the SR, and this is why I did it in two different colors. Calcium comes in, goes to the SR, opens it up, allowing more calcium to come out. This is called calcium-induced calcium release. So calcium coming in, causing more calcium to come out from the SR. This is called calcium-induced calcium release. Now, we have this inactive protein. This calcium goes and binds to comodulin, activating comodulin. Remember when we were learning about protein activators, one of them was a cofactor? Calcium was a cofactor. So, calcium binds to comodulin, activating comodulin. Once comodulin is activated, because now it has calcium, it goes and activates myosin light chain kinase. So myosin light chain kinase is activated. Okay? So comodulin activates myosin light chain kinase. So now we have myosin light chain kinase activated, right? That goes, what is the function of myosin light chain kinase? It takes ATP, converts it to ADP and PI. So myosin light chain kinase takes ATP, converts it to ADP and PI. And this ADP and PI are now placed on this ADP and PI are now placed on myosin. So now myosin has energy. Myosin now has ADP and PI. Myosin now has energy, and it will form cross bridges, and a power stroke will occur. Okay? Does that make sense?
Step 5.
Myosin light chain kinase getting activated. No. Calcium coming in, opening up SR, more calcium coming out. That part is called calcium-induced calcium release. Yes, Jason? Oh, Mark, you had a question? No? Does that make sense? This is smooth muscle contraction.
Are you saying that you got videos explaining?
Well, there's no video on this. Okay, listen up. Let me explain the whole process with this one big slide. So look at my mouse. Calcium from the extracellular fluid comes into the smooth muscle cell by calcium voltage dependent, voltage independent calcium channel. Once that calcium comes in, it rushes to the SR, causing the SR to open up, meaning it opens up the ryanodine receptor. So more calcium comes out. So now the cytosol is flooded with calcium. Where did that calcium go? It binds to comodulin, activating comodulin. Once comodulin is activated, it will grab myosin light chain kinase, activating it. Once myosin light chain kinase is activated, it will break down ATP into ADP and PI. This ADP and PI will be now placed on myosin, activating myosin. So it has ADP, PI, now myosin has energy, it's activated. This activated myosin forms cross bridges with actin, the thin filaments, and a power stroke occurs, which is contraction going on. Okay? This image shows smooth muscle contraction. Okay, question for me. Yes, everybody. So the answer is B. Activates myosin light chain kinase. Okay, we are done with today's lecture. You can turn it off.